Ramshekhar has been really toiling. Poor guy has had the slider is giving him a difficult time.
Yeah, the room is open, sir. I was just going live on YouTube. That is why I was away from Zoom.
No problem, no problem. Mansi,
The flyer we have for Thursday, I'll show that in between.
great. This is the same thing, na? Dr. James Jose?
Yes, even I'm guessing so.
No, you forwarded it. Who sent you that message originally? I don't need to check with Rakesh, correct?
No, sir. I think the Thursday flyer is ready. I think so.
Okay, ready? Okay, okay. You can show. Dr. James Jose, Kozhikode is going to they are going to have a long case on higher mental function, correct? That's the thing, na?
I'll just.
Yeah. So for both of you, Sridhar and Ramshekhar, Ramesh, the lab, the pathology and the metabolic lab, do they support you in any different special way? Is there anything special they've set up? Because
The in-house lab at our hospital, it was not of much help for us initially, metabolic and other tests.
Yeah.
But of late, they are trying to develop few facilities.
Yeah, because you need it, na? Otherwise.
Yes.
Otherwise,
But yeah, luckily we have a good metabolic lab in Sandor Lab, Hyderabad here.
yes.
So far we have been utilizing that. So they have.
They are taking samples from all over the country, correct?
Yes.
Yeah.
So metabolic and genetic tests also they have.
Genetic as well,
Mostly
yeah.
we are using it for metabolic part.
Good. Yeah. We have this NIV in our place. But I don't know, we've not managed to exploit it much really. I've been there a couple of times. I asked them dengue, strains, and Rahul and I wanted to do a little bit on that. Whether the decides which one of them will have this bad thalamic lesion. I don't know. They didn't have that much. They are rather more into day to day. Now with COVID, though, they are not paying attention.
They are fully occupied. Yeah, same for us with CCMB. Not much luck to do any research work or any basic science to collaborate with them. Although it's such a good facility.
I mean, absolutely. What I thought with all that, Uttar Pradesh thing, the hypoglycemic thing, they should have either proven or disproven something,
So don't know what
encephalitis. Yeah. They should have run some viral panel and said that we didn't find any virus. That lychee thing is, I don't know how much strongly, what can I say, we should believe it, or whether it's just a hypothesis. I don't know.
There was a team of people from AIMS who had stayed there for a few days and gathered the data, but we haven't got anything out of that. We haven't known anything.
That's exactly what I'm saying. We have national institutes, but we are not able to prove or disprove anything.
They haven't even done MRIs in all cases.
Yeah, that's really sad. That's sad. Calling it encephalitis without a MRI in today's era. Any WHO or any foreign organization is going to, look down upon us for the quality with we worked up patients. I know. That's really sad. It should have been better. Certainly, it should have been better. Ramshekhar?
Yeah, tell me.
Yeah, everything fine, Ramshekhar? You are
Yeah, so far the order looks okay. The choices look okay. I
good.
just hope they appear correctly online. That's all.
No, no, now everything will be
Uh,
fine, Ramshekhar.
so,
Tell
Dr. Menon, I think you will have to talk a little louder because I mean, on a cell phone, your voice is coming out very low.
Yeah, yeah, I think he will manage to.
Hello? Hello?
Yeah, am I
Ramshekhar?
audible? Am I audible?
Yeah,
Yes, now you are audible.
Okay.
now it's good. It's good. Good. We'll wait for another couple of minutes for people to join. I think people are still in holiday mood. So I left it the last. Some of the PGs have just about joined our PGs, and the old guys, their exams are not yet this thing. Correct? We just managed to finish the July session exams. Correct? Some of them. Difficult times. After you have your January students join, correct?
Yes, sir.
The Jan thing.
Didn't get you.
Mansi,
Yes, sir.
hello. Let's begin. I think. Uh, let's start
Oh,
because
yes.
we have a very good, slightly long quiz also to go. So, to all PGs, good evening. We are back again. And after you've had this two sessions of pediatric neurology, uh, we have we have sort of continued that with lectures on genetics, and today's session will contain two lectures on genetics, as you can see, followed by a quiz, as we do periodically on the topics we've done in the last three sessions. And the sessions would be pediatric neurology and genetics. To begin, uh, obviously, trinucleotide repeat disorders form a very important genetic subgroup that we confront time and again. This particular question has appeared in our exams frequently as well. So we've chosen this one, and that will be followed by epigenetics, which is a relatively new concept, newer than I must say trinucleotide repeats. So let us begin our first lecture on trinucleotide repeat disorders, which will be delivered by Dr. Ramesh Konanki, who is a dynamic neurologist from Rainbow Children's Hospital, Hyderabad. And I request him to begin his presentation. Ramesh. Yeah.
Thank you, Dr. Sripad. I think this is a wonderful platform for the trainees and young neurologists to learn. Thank you for giving this opportunity to share and in the process read as well. So next 15 minutes or so, we'll go through the trinucleotide repeat disorders, which is not common individually, but I think together they make up a quite large chunk of neurological disorders, especially in adult neurology practice and to some extent in child neurology practice. So I would cover these conditions with an overview of repeat expansion disorders. This is the broader umbrella term of which trinucleotide repeat disorders is a subgroup and the largest subgroup. Then pathophysiology and genetics of trinucleotide repeat disorders, that will be the main focus of today's talk session, and spectrum of these conditions, and very little bit on therapeutic advances. The reason being there's not actually much advances in terms of therapy except for one disease. So just to begin with the very basic thing, what are these repetitive sequences? So if we remember our genome, not all the material within our genome is useful for us as such. So there is about 30% of the total human genome which is actually kind of buffered, which means we don't routinely use it in the human evolution, but these contains what are called repetitive sequences. The nucleotides that we have seen in read in our physiology books, these nucleotides, they are repeating multiple times, either in tandem or dispersed in multiple places over the chromosome. So these repetitive sequences scattered in the microsatellite regions most commonly, they account for about 30% of the human genome. So what is the purpose of this when it is not in use? In a normal person, the main purpose of various lengths of these repetitive sequences, which is nothing but DNA, is to allow for evolutionary plasticity. So for a individual person, it may not make much sense, but this is kind of a DNA buffer. When these repeats extend beyond the code of a viable protein, suppose you have a useful functional protein which is transcribed and translated from a piece of DNA, and the number of repeats extend beyond this region, beyond this code for a useful protein, the expression of that particular segment is suppressed. That is the natural protective mechanism that we have. But after a certain threshold number, so if once the repeats keep on increasing, once the threshold is reached, this suppression of transcription is also lost. So what do you have once this suppression is gone? An aberrant protein, which is abnormal protein, which is coded by this increased number of repeats of DNA. And that gives rise to either a functional protein or a non-functional protein. If it is a functional protein, you would call it as a gain of function mutation. Or if it is a non-functional useless protein, you call it as loss of function mutation. So that's how the repetitive sequences can either be loss of function or gain of function, which is also true for other types of mutations. So what are repeat expansion disorders? So this is a large umbrella group of which trinucleotide repeats are one subgroup. So these are group of human diseases which are result of an abnormal expansion of repetitive sequences. So normally we have certain number of repetitive sequences. When there is abnormal expansion in these number of sequences, they result in abnormal protein synthesis and disease. These are repeat expansion disorders. Unfortunately, most of these repeat expansion disorders primarily affect the nervous system, mostly central nervous system, and some conditions also the peripheral nervous system. And they occur during various stages of human development. The DNA repeats come in various sizes from 1 nucleotide to dodecamers, that is 10 repeats, not 10 repeats, 10 nucleotides. Currently, more than 40 diseases are known in humans due to repeat expansion disorders. The first ones described were the trinucleotide repeats. So repetition of 3 nucleotides. That's why they are called trinucleotide repeats. So suppose you have CAG. So these are 3 nucleotides. And again, next to that will be CAG, then CAG. So these are 3 nucleotides which are repeating multiple times. Okay. The size of repeats can range from trinucleotides, that's the largest group. Most of these disorders are trinucleotide repeats, but sometimes you can have 4 nucleotide repeats also. The classic example is type 2 myotonic dystrophy, or you can even have pentanucleotides. So 5 nucleotides repeating every time, 5 sets of 5. That is SCA10 and SCA31. Hexanucleotides and even dodecanucleotides, that is 10 sequence of nucleotides which are repeating successively. So that is progressive myoclonic epilepsy type 1 or Baltic myoclonus. So, but of these, the commonest is the 3 nucleotide repeats. The threshold at which the repeat expansions become symptomatic varies with the specific disease. So some diseases can have threshold of 20 nucleotides, some may have 40, 44 like that. Then little more about the genetic basics of these conditions. If you see, this is the schematic of a gene, say one individual gene. So what do you have? The green is the coding region that is called exon. So it can be either continuous or discontinuous. So you have a bit of coding region that is exon here, another coding exon here, another one here. The intervening period parts of gene which are non-coding, they are called introns. And you have ends of coding regions, 3' untranslated region and 5' untranslated region. So highlighting the fact that the repeat expansions can happen at any part of this gene. So it can be at 3' end, it can be at 5' end, it can be in the coding region, or it can be in the non-coding intronic region. So accordingly, over last 15 years or 20 years, people have identified human diseases in all parts of the gene. So the first box showing Fragile-X syndrome, that is a classical triplet disorder, mainly happening at 5' untranslated region. In the coding exons, we have Huntington disease and some of the spinocerebellar ataxias, spinobulbar muscular atrophy, oculopharyngeal muscular dystrophy. There are intronic diseases as well, not very common, but classical being the Friedrich's ataxia and some of the spinocerebellar ataxias. And at 3' end, not very common, but the commonest is this myotonic dystrophy type 1. So this is just to highlight that different parts of gene can have the nucleotide repeats, which can become abnormal. Again, another point to reinforce, the function of these abnormal repeats can be either loss of function or gain of function of a protein or RNA toxicity when there is a more RNA. So that can vary from disease to disease. Then how do these trinucleotide repeat disorders come into existence, or how do they form? What is the etiology? This is not very clear. This is only a hypothesis. How do they become into existence? There are various environmental factors which have been postulated to trigger cellular responses which result in mechanisms to increase the chance of cell survival. So basically, whenever there is a cell injury due to various environmental factors, always there are adaptive mechanisms to help the cell survive. Whenever injuries like cold, heat, hypoxic injury or oxidative stress happen to the cell, the one effect is to compromise the fidelity of DNA repair. So just remember that keyword, fidelity of DNA repair. So whenever there is DNA injury to whatever trigger, cellular mechanisms try to repair the DNA. How faithfully the DNA is repaired determines whether you will get a good DNA or a bad DNA and the disease. So whenever this fidelity of DNA repair is gone or it is reduced, you will see more mutagenesis or more abnormal DNA naturally, and there's more prone for breakages. So these particular impaired cellular survival mechanisms, they happen mostly in the microsatellite regions. That's where you have a lot of repetitive sequences of DNA. And these regions, for this reason, they are highly mutable, the microsatellite regions, because a lot of replication is going on. And they respond with re-replication in the form of trinucleotide repeats. So just remember those keywords to help you remember what happens with trinucleotide repeats. These are the general features of this group of diseases. Mostly they arise from normally existing polymorphic repeats, which means these are not, to begin with, these are not abnormal. You have normal defined set of repetitive sequences, which are there in everybody, all of us. So they arise from there. The expansions are unstable or dynamic. That's the keyword in genetic understanding of these disorders, which means they don't pass on from generation to generation in a fixed manner or a static manner. They often change size when transmitted to the next generation. And that brings us to the next point, that is, longer the repeats, they tend to cause more severe disease. There's also earlier onset of these conditions. That is called clinical anticipation. Earlier onset, more severe disease in successive generations. And for this reason, because it can be imagined like a spectrum. Suppose if you say for example, any disease, 20 repeats is the normal, or say 5 to 20 is normal, and 40 is the threshold for disease. From 40 to say 200 or 1000 repeats, depending on the number, you can have a large spectrum of disease. That's why the phenotype can be highly variable within the family. And that is due to repeat size itself. Briefly on the epidemiology, Fragile X syndrome seems to be the most common of triplet nucleotide disorders, with a prevalence of about 1 in 4,000 boys with, and this is one of the commonest cause of intellectual disability in males. Myotonic dystrophy and Huntington's about 1 in 10,000. And most of the spinocerebellar ataxias about 1 in a lakh. These are they are little rarer. Friedrich's ataxia, although individually very rare, but that's the commonest autosomal recessive ataxia, accounting for 50% of this subgroup. Together, if you see probably in adult neurology practice, that's a substantial group of diseases. Most of them are unfortunately life-shortening and have debilitating symptoms. And so far, no disease-modifying treatments have been available for this group of conditions. What are the factors that contribute to pathogenesis of these diseases? One, the sequence of repeat itself to begin with, and the size of repeat. Like we have discussed earlier, the higher the number of repeats, more severe is the disease. Sometimes the location of repeat within the gene, whether it's in the intronic region or exonic region or 3' and 5', that can also determine the pathogenicity of the mutation. Whether repeat encodes RNA or protein, that can be a factor. What is the function of repeat containing gene, whether it's a useless non-functional gene versus a functional gene, that can determine, and the extent of meiotic and somatic instability. Some of these things it is difficult to understand for us, but at least some things are straightforward in genetics. So accordingly, you can have toxic gain of function mutations or loss of function mutations. I'll not go through all those individual diseases. Briefly on the modes of inheritance of these conditions. Conventionally, when we talk of Mendelian disorders or single gene disorders, they are typically a fixed mutation or a static mutation within a family, and they cause disease through these patterns, autosomal dominant, recessive, or X-linked recessive or dominant mechanisms. And within the family, usually they have a similar phenotype, maybe with slight variation in the severity, but not too much of a phenotypic variability. That is true for most of the Mendelian disorders. Whereas if you come to trinucleotide repeat disorders, the mutations are very, very unstable and dynamic. That is, the mutations keep changing with successive generations. So as we move from one generation to next, depending on the size of the mutation, they can manifest with markedly varied phenotypes. We'll see some of the examples to understand how starkly different phenotype you can have in the same families. Majority are inherited as autosomal dominant conditions. So most of the classic trinucleotide repeat disorders are autosomal dominant and they have gain of function mutations. Whereas some are autosomal recessive. Classic examples are Friedrich's ataxia and can be X-linked recessive like Fragile X syndrome, and X-linked dominant also rarely, Fragile X tremor ataxia syndrome and spinobulbar muscular atrophy. Only one condition which can have dominant or recessive inheritance is OPMD, that is oculopharyngeal muscular dystrophy. Usually it is dominant but can have recessive inheritance also. Just little bit on anticipation, because this is the most important genetic concept we need to understand with respect to these diseases. This is nothing but tendency for disease severity to increase in successive generations of a family, which is straightforward. We have emphasized multiple times. It is explained by the tendency for expanded repeats to further enlarge upon transmission to the next generation. So with each passing generation, the number of repeats keep on increasing. Also, the longer expansion tend to cause earlier onset, more severe disease. The extent of anticipation and degree to which there are parent of origin effects. So this is another keyword I want you to remember, parent of origin effects. That means depending on which parent, whether it's a mother or father or the male or female which is carrying the repeat expansion, the phenotype will change, or the anticipation can change from disease to disease. For example, there is a significant paternal effect, which means only if these repeat expansions are present in father, they tend to be passed on and tend to become pathogenic. This is true for Huntington disease, SCA2 and 7. Whereas there is strong maternal effect, which means if these repeats are present in a mother's DNA, they tend to cause more likelihood of transmission. That is seen with myotonic dystrophy type 1. So this is what I meant by parent of origin effects. This is true for triplet repeat disorders. One exception for not having anticipation is oculopharyngeal muscular dystrophy. The reasons are not known, but probably it's a very, very small piece of triplet repeats. Then what is the correlation between repeat length and phenotype? Again, there is in most of the diseases so far what we have understood, there is a striking genotype phenotype correlation between repeat length and the disease severity. Longer the repeat, more severe disease and earlier onset of symptoms. So this is straightforward, except the OPMD. Differing sizes of the same repeat fundamentally alter the way sequence behaves and affects the nervous system. So just to give you an example, this is the phenotypic spectrum of family with spinocerebellar ataxia type 3. So if you see the number of repeats here, so this is the normal number of repeats in non-affected individuals, probably up to 40 repeats, 12 to 40 repeats is normal. So if you have maximum number of repeats, say 80 or 100 repeats, they manifest with predominantly dystonia. So that is called type 1 SCA3 with early onset with dystonia. So they are probably younger individuals. Whereas if the number of repeats are less than that, they usually manifest with progressive ataxia, midlife onset with progressive ataxia and brainstem signs. Whereas even lesser number of repeats, they just manifest with late life onset, probably 4th, 5th decade, with the distal leg weakness due to neuropathy, motor neuronopathy, and milder ataxia. That's the milder phenotype with very late onset. So earlier onset, more severe phenotype, lesser severe, and least severe phenotype. So these three phenotypes are very well known. But of late, there are cases of even lesser number of repeats, but more than the threshold, we're presenting with restless leg syndrome. That is given type 4 SCA3 with intermediate expansion. So this is borderland between the normal repeats as well as definitely pathological. So that's another phenotype. So this is a classic example for trinucleotide repeat disorders and the varying phenotype. So this is the large group of relatively well delineated diseases due to trinucleotide repeats. I'm not going into individual conditions. I'll briefly go through 4 commonest diseases which we know. First one is the Friedrich's ataxia. This is an autosomal recessive disorder involving GAA triplet repeats. So here it is GAA, and the gene is FRDA on chromosome 9. This gene encodes a protein called frataxin, which is located in the mitochondria and severely reduced in these patients. So what are the salient neurological findings? This progressive gait and limb ataxia, areflexia, especially in the lower limbs, but with extensor plantar response. So you have pyramidal as well as neuropathic signs, dysarthria and sensory neuropathy. That is the neurological phenotype. But many of these patients also have cardiomyopathy, scoliosis, and foot deformities, again part of neuropathic weakness. So this is the classical phenotype of a Friedrich's ataxia. Huntington's disease due to CAG expansion, pathological expansion of CAG repeats, and mostly it is due to paternal transmission. That is again the parent of origin effects. What is the clinical triad? One is the progressive movement disorders, most commonly chorea, progressive cognitive decline, later age going into dementia, and various behavioral disturbances that often precede diagnosis and can vary depending on state of disease. So I'm not an adult neurologist, so probably adult neurologists would see much more commonly. Again, depending on the number of COG repeats, you can have a asymptomatic individual or milder phenotype, what is called Westphal variant, which is a younger age of onset, and the basis is the number of repeats. Myotonic dystrophy, this is another condition which can span whole of life, starting from neonatal period to 40s and 50s in terms of age of onset. As you can see, there are variants which start at birth with the severe hypotonia, respiratory failure, not able to feed, and cardiorespiratory complications. That is the severe most phenotype. And as you can rightly see, the CTG repeats, so those are the nucleotide repeats, are usually more than 1,000. So they have the severe most phenotype. Whereas milder ones, childhood onset, you can starting between 1 to 10 years, mainly present like a myopathy, congenital myopathy. You have facial weakness and myotonia. And the juvenile variant in the first and second decade, you have motor and cardiac symptoms can sometimes be absent. Adult onset 20 to 40 years, again weakness, myotonia, cataracts, conduction defects in the heart, endocrine manifestations. These are all much more common in adult onset cases. You have late onset or asymptomatic cases also with just very mild myotonia and cataracts, and the premutation, obviously they are asymptomatic. So this is a very widest phenotype that we can have, starting from birth to 40s and 50 years to have an onset of disease. Fragile X syndrome, which even we don't see very often because the phenotypic features or the facial features are little non-specific in younger age, but as they grow into second decade, the facial features become more prominent and recognizable. And this is the commonest genetic cause of intellectual disability in boys. And this is due to CGG repeats in the Fragile X mental retardation 1 gene, FMR1 gene. And it has 4 forms depending on the number of repeats. Normal below 40, intermediate 41 to 60, up to 20 pre-mutation, and full mutation is more than 200 to 230. And the most prominent one are the facial features. They have typically long face, very prominent ears, prominent jaw, macrocephaly, macroarchidism. So that's the usual facial dysmorphic features that you can make out, typically in a second decade, older child. They have behavioral problems in terms like ADHD, anxiety, autism spectrum disorders. And it occurs almost exclusively through the maternal transmission. Friedrich's ataxia. Okay, sorry. So I have covered Fragile X syndrome, Friedrich's ataxia, myotonic dystrophy. These are the common conditions, and Huntington disease. So just briefly to give you an overview of the common diseases and how does the phenotype changes with within the same families. Briefly on the treatment, I think only condition which has been well researched and still the trials are underway is Friedrich's ataxia. All other diseases, the symptomatic and supportive care has been the key mode of management. So some research on Friedrich's ataxia, a lot of antioxidants have been tried, including CoQ, vitamin E, idipinone, N-acetylcysteine, and hormonal therapies like dihydroepiandrosterone, proglitazone, PPR gamma inhibitors, and deferriprone and iron chelator has been shown to prevent buildup of reactive oxygen species. And erythropoietin has some role in brain's response to neuronal injury and cell repair. Interferon gamma 1b increasing the frataxin expression in dorsal root ganglions, and HTACs inhibitors. So these are all experimental. Most of them have shown some improvement in terms of basic science parameters, but actual clinical parameters, there has not been much of a progress. Some of the experimental therapies, mainly 2 groups of experimental therapies. One is what is called exon skipping through antisense oligonucleotides. Mostly in the preclinical stage, if you see, most are in this stage. Most of the trials for Huntington's disease, SCA 1, 2, 3, 6, 7, and myotonic dystrophy. Most of them are in preclinical phase. And the other therapies are vector-based gene delivery therapies using typically adenoviral associated vector gene therapies. So far, nothing in the human clinical trial phase. Mostly these are preclinical stages. I think that's about trinucleotide disorders. The first one is has been very useful resource from PubMed, and other two articles are also quite useful for your reviews. Thank you.
Thank you, Dr. Kumar. I think it was very concise, to the point, and I think very nicely you showed everyone how to highlight the important ones amongst the large list of disorders. The best part was, I think, myotonic dystrophy, taking it as an example and showing how the spectrum could be so wide. Great, thank you. Let's move on to our next lecture, as you know, on epigenetic disorders by Dr. Sridhar, who is a young neurologist from SKS Neurocenter, Hyderabad. I request him to enlighten on this relatively new concept. Sridhar.
Good evening, everyone. I thank Dr. Shripad and Fine for giving me an opportunity to speak on this platform. Today, my presentation is on epigenetics in neurology. Epigenetics involves heritable changes that alter gene expression without altering primary DNA sequence. And neuroepigenetics is a study of how epigenetic changes to genes affect the nervous system. And these epigenetic changes are heritable provided these changes occur in the germ cells. Now, this cartoon summarizes the various epigenetic modifications. These epigenetic modifications are responsible for the various specialized cell types in our body. They're responsible for individual variations in the monozygotic twins. They're responsible for various stages of development from the embryo to newborn. They're responsible for the various disease manifestations based on the position in the genome, the various functions in our body, and transgenerational manifestations of diabetes, obesity in response to the environment. And they're responsible for a number of neurological, hematological, and disease and oncological disorders. Now, for the computer geeks, what does this mean? Like, if you're a computer geek, genes means computer hardware and epigenetics means computer software. And for people who are voracious readers, genes means words and epigenes means how the words are read. Now, this cartoon shows embryo which differentiates into various cells like the blood cell, the nerve cell, the bone cell, and the skin. And this differentiation is brought about by epigenetic mechanisms which can be reprogrammed, which are malleable, which are inheritable, and which can be reversed. This slide summarizes the differences between epigenetics and genetic mechanisms, and epigenetics varies from one cell to another, whereas genetics remains constant across these various cell types. This can be reprogrammed, whereas the genetics cannot be reprogrammed, and changes made are reversible, whereas the changes made in genetics in the genes are not reversible, except for gene editing techniques like the CRISPR-Cas9 and Cas13. Now, these are the various basic terminologies used in epigenetic disorders. Back. Okay, so epigenome consists of the entire epigenetic code across all of the cells in the body. Epigenetic modifications involves chemical alteration, that is covalent attachment, methylation, and acetylation to the chromatin or the DNA that does not affect the primary DNA structure. Gene silencing involves reducing or completely suppressing expression of gene through epigenetic mechanisms, and imprinting involves specific epigenetic modifications that are only added in either the male or the female gametes, that is, they are parentally imprinted. The Prader-Willi syndrome has paternal imprinting, whereas the Angelman syndrome has maternal imprinting. Now, few words regarding the epigenetic regulation. It's an additional layer of gene regulation beyond the DNA sequence. It involves more combinations and hence larger variety of outputs, potentially more extensive than the routine genetic regulation. It is more malleable and programmable and reversible. Now, this cartoon shows, the first cartoon here shows where the DNA transcription as well as the translation process, when the DNA is converted into the RNA, and the RNA, once it comes out of the nucleus, it is converted into proteins by the translation process. And this one summarizes the how compactly the DNA double helix model of DNA, which is 3 meters in length, is compactly arranged in a nanometer nucleus. And this one summarizes switching genes between the on and off states. The on and off states occur when the histones, that is the DNA double helix, is wrapped around these histones. And when these histone bodies, histone proteins are very close to one another, then the genes are silenced and they're not available for transcription. But when they move away from one another, then the transcription process is quite possible. Now, how this is brought about is through the modifications. There are two modifications. One is in the DNA and the other one is in the histone protein. And these chemical modifications were described as way back in 1964, and most of them occur in the histone tail. Based on whether it occurs in the distal, the middle, or the proximal portion, the various diseases can do occur. And how these modifications are brought about was discovered in 1996 these modifications are brought about by various enzymes known as the writers, the erasers, and the readers. This is the heterochromatin, where there is no transcription and the histone proteins are very close to one another, and the DNA is compactly arranged around these proteins and is not available for transcription. And histone acetyl transferases move these histone proteins further away from one another, and the DNA is available for the transcription process. The various type of epigenetic marks which are already emphasized, that is the DNA methylation and histone modification. There is one more epigenetic mark, which is a higher order chromatin modification, though this is not classical. And the various physiological processes that are regulated by epigenetic mechanisms include the X-chrome inactivation or the lionization, the cellular differentiation, and the generational onset of adult onset diseases like diabetes and obesity in response to the environment. This cartoon summarizes the various epigenetic marks. Here we see the histones, and these are the histone tails, and the DNA is closely, compactly wound around these histone proteins. And this is the histone modification site, and this is the DNA modification site. Now, the DNA undergoes methylation by DNA methyl transferases. There are 1, 2, 3A, 3B, and L types of DNA methyl transferases. These are known as the writers. And once they are methylated, these information is read by the that is readers, that is the MBD proteins. And similarly, the histones undergo a number of modifications. Usually there are a number of modifications, but the most common types are the 5, which can be summarized by a non-approved acronym AMPUR. A stands for acetylation, M stands for methylation, P for phosphorylation, R for ribosylation, and U for ubiquination. And once this is acetylated, these are known as the writers, which acetylate the histones. And the histones, acetylated histones, this information is read by the readers known as the bromo, chromo, Tudor, PWT, PhD finger domain proteins, which further pass on the information for the transcription process. Now, most of the methylation in the DNA, on the DNA, occurs at the 5th carbon atom on the cytosine and in the promoter regions. And these are the epigenetic marks on the DNA. Well, now coming to higher order chromatin modifications, these are basically the LOX and the LADs. Now let's look at this cartoon. This is the inner nuclear membrane. This is the nuclear lamina, and this is the lamina associated domain. And the green dots here, they are the LOX, that is the large organized chromatin lysine K9 modifications. Now, hypomethylation brings about separation of these from the nuclear lamina, and this causes the euchromatin marks to come closer to one another and undergo recombination or break up to form DNA breaks. And this leads to silencing of a large number of genes or overexpression of large number of genes. Now, the various physiological processes regulated by epigenetic mechanisms include the X chromosome inactivation, cellular differentiation, and transgenerational adult onset disease like diabetes and obesity in response to the environment and the aging process. The role of various epigenetic changes in the disease involves the developmental disorders and the environmental effects on the health, cancer, and a number of neurological disorders. Among the developmental disorders, the most well-studied are the Prader-Willi syndrome, which is paternally imprinted, and the Angelman syndrome, which is maternally imprinted. The other syndromes are the Rett syndrome, the Rubinstein-Thiaby syndrome, the Coffin-Lowry syndrome, and the ATRX syndrome. I'll be concentrating mainly on the neurological aspects of the epigenetic changes that affect the disease, that affect these neurological diseases. Now, the prototype for almost all the neurological disorders, as far as epigenetics is concerned, is studied mostly in the glioblastomas and the neuroblastomas, that is the neuro-oncology department. And this involves, that is the epigenetic and the epigenetic alterations are involved in cancer initiation and progression. This is brought about by global DNA hypomethylation and specific CpG island hypermethylation in tumor suppressor genes. And once these genes are silenced, the regulation of cell proliferation, DNA repair, cell differentiation, and other homeostatic functions are affected. And this evades the regulation of the cell cycle and the cells become tumorigenic. There are a number of comprehensive CpG island hypermethylation profiles for GBMs, that is glioblastomas and neuroblastomas. The DNA repair gene MGMT is a biomarker of clinical response to alkylating drugs in gliomas. Now, the prototype for neurodegenerative diseases includes the late onset Alzheimer's disease, which is caused by reduced clearance of A-beta amyloid. There are a number of mechanisms which are responsible for the A-beta amyloid accumulation, neprilysin causing hypermethylation in the cerebral endothelial cells and histone acetylation in the MB7 cells, the AICD interactions with the histone acetyl transferases causing acetylation and phosphorylation leading to a H2X accumulation, which is an indicator of DNA strand break, the S100A2 DNA methylation, SORB S3 DNA methylation. In Parkinson's disease, TNF-alpha DNA hypomethylation and histone hypoacetylation leads to TNF-alpha overexpression, which induces apoptosis in the substantia nigra cells. In Huntington's disease, interactions of the mutated Huntington protein leads to hypoacetylation and H3K hypertrimethylation, which leads to caudate and cortical neuronal death. In ALS, the fusion inhibition of the histone acetyl transferase activity leads to hypoacetylation of histones and aggregates of SOD1, that is superoxide dismutase 1, TDP43, and fusion proteins, which leads to cell death. And histone deacetylases type 4 is dysregulated in ischemic stroke, and this plays a key important role in stroke as well as post-stroke recovery by affecting neuronal death, angiogenesis, and neurogenesis. This slide summarizes the various DNA methyl alterations occurring in various diseases and the impaired pathways. Most of them have already emphasized, and multiple sclerosis is caused by hypomethylation, and the impaired pathway involved here is the myelin basic protein citrullination. This slide also summarizes the histone alterations which occur in various diseases and the impaired pathways. Now, coming to the therapeutic uses, there are a number of drug classes for epigenetic modifications to occur. The most important drug classes are the hypomethylating agents and the histone deacetylase inhibitors. The main disease targets are as below, but I'll be concentrating mainly on the neurologic and the psychiatric disorders. Now, therapeutic drugs act by two broad mechanisms, directly on the epigenetic marks and indirectly by bringing about changes in these marks. The hypomethylating agents, the drugs that inhibit DNMTs, that is DNA methyltransferases, reduce methylation and lead to gene overexpression. The prototypes of these drugs are 5-azacitidine and D-citabine. Histone deacetylase inhibitors bring about histone acetylation and increased gene expression. The main HDAC inhibitors for clinical use include Friedrich acid, trichotomic acid, and valproic acid. Now, this slide summarizes the various mouse models tested in the phase 2 trials, the type of drug and the effect it has had on the diseases. Now, let's concentrate on ALS, wherein valproic acid, which acts as a HDAC inhibitor, helps in disease onset delay and lifespan extension. And similarly, AZA and SAHA, which are DNA methylation inhibitors and HDAC inhibitors, help in restoration of neprilysin mRNA expression and increase the number of synapses and help in memory facilitation. In multiple sclerosis, trichostatin A, which acts as a HDAC inhibitor, reduces demyelination and neuronal loss. Now, these are my last summary slides. Epigenetic modifications involve changes to DNA or histones without changing the primary sequence of the DNA. DNA can be the 5-carbon atom of the cytosine in the mammals in the promoter region. Histones undergo a number of modifications like acetylation, methylation, phosphorylation, ubiquination, and ribosylation. Epigenetic changes are heritable and programmable in response to the environment or development. Normal physiological processes affected by epigenetics include lionization, cell differentiation, and adaptations to the environment. Prader-Willi and Angelman syndromes are disorders affected by parental imprinting. Epigenetic changes in response to environmental exposure can be associated with adverse health conditions, and DNMT and HDAC inhibitors are used in cancer treatment and hemoglobinopathies. As of now, only 4 drugs, epigenetic drugs, have been approved approved by the FDA for treatment, and these are mostly for some solid tumors and hematopoietic stem cells, hematopoietic tumors. These are my references, and thank you for the patient hearing.
Fantastic. I have to say, Sridhar, you made this very difficult topic so look so easy through your so fluid and teaching type of a lecture with all the flow chart like method, the way with which you taught it. Very nice. I really like this lecture. I think this is very new to at least to me. Excellent. Thank you very much, Sridhar. Just before we move on to the quiz, just a short reminder for Thursday, you will have a higher mental function case presentation in front of Dr. James Jose, the professor and head of neurology at Kozhikode. And kindly take advantage of this session on Thursday. And with that, we move on to the last part of today's teaching with a quiz by Dr. Ram Shekhar Menon. You've already heard him give a very elaborative but a concise lecture on neuroregression. He's now going to teach us through quiz, rather revise us through quiz, several topics which we have already covered in the last 3 sessions of Ram Shekhar. Today we are going to have a quiz on poll rather than Slido. He's going to explain us the method of poll as well. Ram Shekhar, after you.
Thank you. Am I audible?
Yes, you are.
Okay. So I think I need to be made the host first.
Yes.
Could you?
Manasi? Hello? Manasi? I think she's around. Manasi? Hello? I'll just call her. Maybe she's in the middle of another. Manasi? Hello? Just hold on a second. Your thing? Ram Shekhar? Yeah, you can share your screen. I don't know whether maybe. Manasi? Hello? Manasi? Hello? I don't know what has happened to her.
No, I can't create the poll, I think, unless I make the
Yeah,
host.
unless you are a, I know that. No, I'm trying to. Hello? Mansi, yalla Ramshekarla, host one. Okay. Ramshekhar, she'll do that in a second.
Yeah.
Yeah. So I think you can start telling them about pool, about how the pool thing works, because this is the first time we are going to do it on pool rather than on the slide.
Okay, so what I'll be showing you is I'll be presenting to you those questions, and once I give you the options, I'm going to enable the poll, which will give you a probably give you a 20 to 30-second window to vote your and give your options. And then I'll share with you the results of the poll. Once you have shared, you just need not do anything, just press your options and just wait for the polling to stop. And then I will be sharing with you the results. And then we will discuss what should be the thinking process behind. I'm sure most of you will find it easy. The quiz has been defined to give a rather clinical weightage to it. So we are starting off with more clinical questions, and towards the end, it will be more theoretical questions, which are directly related to the subject that you must have, the subjects which you have attended. So I don't think you would find it very tough, and unfortunately we can't make this interactive, but I think I'll do my best to probably just tell you about how you could come to the correct conclusion and some theory behind the correct options. So yeah, so I think we'll start off with the first, and let me just go on to the question. So this is the first, I'm just taking the first question. And what I'll do is, so this is the question. So you have a 12-year-old male who was born of non-consanguineous parentage, who presented with progressive imbalance, seizures, dysarthria, followed by executive and episodic memory dysfunction, with significant scholastic decline since the past 2 years. Examination of this child revealed regression in primarily the fine motor and language abilities, including the writing skills, with evidence of a prominent action myoclonus and cerebellar ataxia. I'll be showing you the investigations, what you would need to answer on as to what is the likely possibility. So as you can see from the MRI, you can see that this patient's MRI shows evidence of a cerebellar atrophy. You can see that probably there is a thinning in the body of the corpus callosum. And look at the EEG findings. I'll not disclose to you the EEG findings, but just look at the EEG findings. This is noted at photic stimulation. And on your bottom left is the somatosensory evoked potential response. Look at the N19 response. I'll give you a value of around 20 microvolt. And this is the skin biopsy, which is self-explanatory, the hematoxylin eosin stain, followed by the PAS positive stain and the Lugol's iodine stain. So I think I have given you enough clues. So these are the options. And coming to the poll, I'll just make it enable so that all of you can vote. Oops. So just give me a minute. So I'm going to launch the polling and all of you are expected to vote now.
So what I would request you to do is also do the check of allow panelists to vote so that even we will be able to see it.
Okay, you want me to do that?
Yes.
Okay. I'll just relaunch polling. Allow panelists to vote. Yes, I have enabled that. So I think all of you can vote. I think with the permission of the panelists, we'll probably give 20 to 30 seconds. I can see that only 20 out of 35 have voted. Just one option or the other. Should I assume that the rest of you are not interested in voting, or shall we proceed? We need 14 more votes so as to ensure that all the attendees have participated.
Sir, I think with a time span of 20 seconds, we can move ahead. At the top, I think you can see the number of seconds.
Okay. So I'll be sharing with you the results. I think overwhelmingly majority of the people who have voted for Lafora body disease, while a small subset have voted for subacute sclerosing panencephalitis, neuroaxonal dystrophy, and Niemann-Pick's type C. So as you can see, the correct answer is Lafora body disease. Now, basically what I showed you on the MRI is just primarily cerebellar atrophy. And somebody who developed an appendicular, an action myoclonus is primarily, you are referring to a subject who had neuronal regression with ataxia, as well a regression not only the motor milestones, but also in the language milestones, along with ataxia and action myoclonus. So, SSP, now it remains a possibility in a child who has regression with myoclonus, but the myoclonus is typically a slow myoclonus and it is predominantly an axial myoclonus rather than an appendicular myoclonus. And additionally, there are a few key investigations which I showed you. One was the EEG. So the EEG, I don't know if it was very clear to you, but the EEG had shown photosensitivity, a photoparoxysmal response. The somatosensory evoked potential, I mentioned to you a value of 20 microvolt. Anything more than 8 microvolt is considered to be evidence of a giant potential on median somatosensory evoked response. And the skin biopsy had initially shown you the typical positive iodine staining inclusions in the apocrine set glands. So basically, this refers to a specific kind of a neuronal regression syndrome, which is due to a gray matter disease. So what pointers indicated a gray matter disease? Primarily the neuronal decline in accompaniment with seizures along with myoclonus and the neuronal regression. And if the patient had coexistent spasticity or had spasticity at onset, you would thought of a white matter disease. But here it is predominantly a gray matter disease in view of the neuronal regression with action myoclonus as well as seizures. And this is confirmed by the MRI findings of cerebellar as well as corpus callosal atrophy. So this is a disorder which commonly affects children as well as young adults. And it is a distinctively progressive disorder with no definitive treatment available. But you have certain PME syndromes such as the neuronal cerebral lipofuscinosis type 2, for which certain intraventricular enzyme replacement therapies have become available. But we know that we are in the era of gene therapy, so it is possible that genes which interact or genes which cause this particular disorder, such as the laforin gene or the EPH gene, you might find a gene therapy for this disorder soon. So coming to the next question. So this is a 2.5-year-old girl born of a third-degree consanguineous parentage. Please note the child had initial global developmental delay with a developmental quotient of 67%. And at 1.5 years of age, the parents noted regression in the gross and fine motor milestones, in addition to paroxysms of prolonged episodes of uprolling of eyeball with retained awareness. The prolonged episodes of uprolling of eyeball. Examination revealed episodes of paroxysmal tonic upgaze with encephalopathy with no correlate on EG. So by encephalopathy, the child was not feeding very well, would remain in the state of paroxysmal tonic upgaze. Intrictal examination revealed a developmental age of 13 months. And appendicular dystonia was evident on action with truncal dystonia. And what was prominent during the admission was the presence of significant diurnal fluctuations. And additionally, the child was noted to have foot deformities and a striatal toe. I'm giving you a bit of a leeway here by telling you that supplementation of levodopa led to an excellent response, and she continues to remain well. Her developmental age at last available follow-up at 6 years of age was 70%. The MRI and the EEG were also normal. What is the next set of tests you would like to order? So these are your options. I'm just going to enable the polling. You can vote now. Okay, so I'll be ending the polling now, and I'll just share with you the results. Most of you have voted for genetic testing clinical exome, while a significant subset have also voted for CSF neurotransmitters, and the remaining for the Wilson's workup with serum copper and ceruloplasmin, as well as mitochondrial workup. So these are the results. As you can see, majority of you have voted for clinical exome sequencing, which is quite agreeable and acceptable. And what do you think this disorder is? So this disorder is primarily a CSF neurotransmitter disorder. So it's a CNS neurotransmitter disorder. So what are the pointers to that? So the patient initially had a movement disorder. When I mentioned that the MRI and the EEG was normal, and these episodes of paroxysmal tonic upgaze, you know that you're dealing here with an intermittent extrapyramidal syndrome, along with some signs of a chronic extrapyramidal syndrome in the form of an axial and appendicular dystonia. Diurnal fluctuations indicate the fact that you could think of a dopa responsive dystonia-like condition. But certain neurotransmitter disorders of childhood, which are associated with global developmental delay, may be followed up by developmental regression, primarily in the motor milestones and often in the language milestones. And these patients can develop significant extrapyramidal signs. Often they may have associated ataxia, tremor, Parkinsonism, which is often suggestive of a secondary Parkinsonism, because the Parkinsonism may be more axial than appendicular and may often involve the bulbar musculature or even the supranuclear gaze centers. And what is predominant is that this episodic symptoms in this group of children can be due to an episodic hyperphenylalaninemia. So if they are left untreated, many of these patients can develop a progressive neurological deterioration. And both responses C and D are correct because of the fact that now we are in the era of next generation sequencing, and many of you can understand that with a dystonia plus panel, you can detect many of these neurotransmitter disorders. And before we had a clinical exome sequencing available, we had to rely more on levodopa trials. So none of these investigations were done in this child, is what I understand. And nowadays, if you have the lab facilities available, you can also proceed with CSF bioamine assays. And these bioamine assays should cover those neurotransmitters transmitters which are involved in the dopa metabolism and dopa synthesis pathway. So these include HVA, HIA, and then urine neopterin and CSF neopterin. And if you have the facility, enzyme assays are also available to look at specific enzymes and the pathways which are involved in dopa synthesis. So the primary etiologies include the tetrahydrobiopterin synthesis, disorders leading to hyperphenylalaninemia, and this can be secondary to PTS deficiency, sepiapterin reductase deficiency, and aromatic acid decarboxylase deficiency. I understand of late that this child was later on proven to be having a sepiapterin reductase deficiency, which can explain the very prominent response to levodopa as noted in this child. So this is the next question. This is a 10-year-old girl who was born of a non-consanguineous parentage who presented with initial language delay with intellectual disability, followed by nocturnal seizures, which were predominantly focal motor with impaired awareness. So seizures in sleep. This led to decline in the language, which was both in the expressive and receptive language, along with scholastic abilities since the past 1 year, followed by autism, social withdrawal, and bowel bladder incontinence. So this is a typical childhood disintegrative disorder. Seizures were resistant to polytherapy. Clinical examination revealed presence of coarse facies, bushy eyebrows, macrocephaly with an orbitofrontal circumference of occipitofrontal circumference of 61 centimeters, loss of fine motor skills, receptive and expressive language delay with autistic stereotypies, drooling, and no other neurological deficits. MRI brain revealed non-specific white matter hyperintensities with few callosal cysts. And I'll be just showing you the sleep EEG. The question for you is, how would you like to manage this child? So this is the EEG, this is a sleep record, and just for the benefit of the audience, I'm telling you that this is showing a continuous, near continuous activation of spike and wave discharges in a multifocal pattern with very poor sleep structures, right? So this is the question. How would you like to manage the patient? I'm just enabling the polling. Okay, please vote. It does seem that progressively a fewer number of participants are voting. I would like to encourage all of you to vote. Okay, I'll share the results. So a majority of you feel that we should be aggressive, start managing with corticosteroids, or you can attempt a high dose of benzodiazepine and ethosuximide. A significant proportion also wanted to conduct further investigations in the form of plasma amino acid and urine organic assay. And a small subset wanted to perform urine for glycosamine glycans. So what are the clues here which I had given you earlier? So one thing is the child had prior intellectual disability, but prominently I also mentioned to you presence of macrocephaly, bushy eyebrows, coarse facies, which indicates that this probably had some syndromic basis. So it is obvious that the deterioration in the child was due to an epileptic encephalopathy, that is CSWS, and you are tempted to initiate a trial of corticosteroids as well as attempt whatever, but you are not very, you know that this is not an idiopathic epileptic encephalopathy. This is not a prototype developmental epileptic encephalopathy which has an idiopathic basis. You're not talking about a completely normal child who went on to develop a language regression. You're talking about a child who has intellectual disability as well as some features of dysmorphism. And that is what should prompt you to think that probably you have a symptomatic basis. A symptomatic basis, what I mean is it could very well be a genetic basis, it could be a structural basis, or it could be an inherited metabolic disorder. Now, one of the disorders which is known and which is very typical of the dysmorphias which were associated in this patient, that is the macrocephaly, the bushy eyebrows, the coarse faces, is mucopolysaccharidosis. And this child was found to have Sanfilippo syndrome, which is mucopolysaccharidosis type 3A. And just treating the epileptic encephalopathy without knowing the etiology does not really make sense. Uh, while it is good that in the intervening period you can initiate corticosteroids, you can do ethosuximide, you can do benzodiazepines and whatnot, uh, the patient may not respond to treatment. And this patient surely did not respond to treatment with corticosteroids and whatever was tried. And because we were suspecting a symptomatic basis, this genetic testing was ordered. So you could either order urine for glycosamine glycans, which is probably the correct answer in this particular situation, whereas genetic testing is also fairly feasible. And this disorder is known to be associated with severe neurological deterioration. Now, when I mentioned that the patient had a severe degree of neuronal regression, not only in the fine motor skills, but also in the language milestones, to the extent that the patient became double incontinent, you must note that you are dealing with a severe childhood disintegrative disorder here. And the childhood disintegrative disorder had an underlying syndromic basis. And just treating the epileptic encephalopathy alone will not change the outcome. Now, unfortunately, Sanfilippo syndrome does not have an enzyme replacement treatment available. So people can argue, we are just trying to treat the developmental epileptic encephalopathy. If the etiology does not have any basis, then what is the point in investigating further? And why shouldn't we focus on the epilepsy? You must understand that when you are initiating that treatment, the patient's family has certain expectations, and unless they really understand that you are likely unless they really understand what is the etiological basis behind the developmental encephalopathy, they are going to have high expectations when you offer them this treatment. So this patient continued to deteriorate despite whatever treatment was given, and which is why genetic testing came out and was confirmatory for this mucopolysaccharidosis, which is a lysosomal storage disorder. Primarily, it's a prominent storage disorder. Presence of macrocephaly, facial dysmorphism in a child who is developing a childhood disintegrative disorder, you should think of a lysosomal storage disorder such as mucopolysaccharidosis. So this is the next question. This is an 11-year-old girl who was born of non-consanguineous parentage with no prior antecedent illnesses, presented with behavioral issues of 3 months duration, with a subacute course of progression over the next 3 months. So she was noted to develop initially inattention at school, followed by loss of calculation skills, poor handwriting, followed by social disinterest, insomnia, inappropriate laughter, prominent mobile and movie addictions. And when she presented, she had developed social bladder incontinence. Examination revealed normal gross motor and fine motor skills, as well as expressive and receptive language, with significant decline in social skills and peer interaction. She was noted to be inattentive as well as incontinence on admission, with obsessive compulsive traits. An MRI did not reveal any structural abnormalities, and an EEG with a sleep record were normal. What do you think is the likely etiology? So I'll just enable the polling. You can vote. Now, okay, I'll move on. So I'll just share with you the results. So a majority of you have rightly thought about pandas, while a small subset have thought about Klein-Levin syndrome, KLS, or Klein-Levin syndrome. And some of you feel that this could still be just Tourette syndrome requiring psychiatric management. So I wouldn't say that you are wrong in terms of thinking about Tourette syndrome, but overwhelmingly the child was essentially normal. So now why are you thinking of an autoimmune basis here? So now pandas, as you understand, is a post-streptococcal autoimmune neuropsychiatric disorder, isn't it? So the full form is actually pediatric autoimmune neuropsychiatric disorders associated with streptococcal infection. And the other terminology that is used is PANS, that is pediatric acute onset neuropsychiatric syndrome. It's also called childhood acute onset neuropsychiatric syndrome. So now this is a dubious entity in itself, but what has been increasingly shown, or what there is a general consensus, is that you have certain children who were developmentally normal, cognitively normal, scholastically normal, who for no specific reason develop obsessive compulsive traits which are of an acute to subacute onset without any antecedent or definite well-defined antecedent. While in some there may be a history of some fever or sore throat, but in general you may not get any definite history of antecedents. And what is very prominent is the presence of these obsessive compulsive traits with social decline, to the extent that, as you were seeing in this particular child, the child had developed urinary incontinence. So in such a situation, when you have a social decline in a child who is essentially normal, with relative preservation of the motor domain, as well as to a large extent the language domain, including the scholastic, and when we assessed, as we mentioned, the reading writing skills look to be fairly appropriate. It was the isolated social decline domain. But here the main red flag was the presence of incontinence and the absence of definite tics, which would favor a Tourette syndrome. So which is for this reason that the child was thought to have a PANDAS or a PANS. Now this child subsequently, as you can see, was investigated, was found to have a normal ASO. So not that is considered to be the signature of PANDAS. This is a raised ASO beyond 250. In this case, the ASO was normal. But because of a high index of suspicion, an anti-DNase-B level was done, which is also considered to be a confirmatory test for post-streptococcal infections. And this was significantly high. And the child is, dramatically responded to a trial of IVIG. So why is it not Klein-Levine syndrome? A large proportion of you felt it is Klein-Levine syndrome. So primarily, Klein-Levine syndrome is characterized by reversible episodes of excessive sleep, and they are recurring to the extent that they go like in terms of rapid cycles of prolonged sleep followed by cycles in which the sleep pattern normalizes. And each time they wake up from sleep, they have this behavior disturbances with prominent hyperphagia, often sexual disinhibition, and disorientation, as well as prominent hallucinations. So this child did not, while the child had definite sleep disturbances, the child did not have this pattern of cycling and did not have any periods of hypersomnolence. This child had only predominantly insomnia. And the child did not have any gross, changes in her eating habits. So while Klein-Levine syndrome is a good thought, the syndrome is, the evolution of this child was not very prototype for a Klein-Levine syndrome. And in Klein-Levine syndrome, both an idiopathic basis, which is believed to have an HLA association, has been described, but certain symptomatic forms, which include autoimmune disorders, tuberculosis, meningitis, hydrocephalus, hypothalamic gliomas, have all been described to be associated with Klein-Levine, but the child's clinical presentation was not consistent with Klein-Levine syndrome. As you are aware, acute intermittent porphyria patients present with acute abdominal pain, vomiting, constipation, and when you test, they are noted to be hyponatremic. They may have seizures, limb weakness, and neuropathy, and they are very often acutely encephalopathic. And this child did not have any features of acute encephalopathy at presentation. So this is Dr. Sripat's question, and I think it should be pretty simple since you have attended this current lecture. So which of the following is an example of an epigenetic inheritance? I'll just enable the polling. I think you should ace this. All of you have repeatedly heard about methylation and acetylation. Yeah, it does seem that overwhelmingly all of you feel that it is because of histone methylation patterns. So I think that is self-explanatory. I'll not spend time on this. So DNA methylation patterns, that is primarily associated with in neurology. As far as we are concerned, we know that it is associated with certain forms of developmental encephalopathies such as Angelman syndrome as well as Rett syndrome. And basically, the mechanism of histone methylation is already fresh in your mind, so I'll not be going into the details of this. So this is the next question. So commonly, what of histones leads to silencing of the genes? So basically, what process, what epigenetic mechanism would actually lead to silencing of genes? The options are phosphorylation, methylation, acetylation, ubiquitization, and all of these processes would silence the genes. I think this is an important theoretical question. Your options are, so please start voting now. Okay, so while a majority of you have voted for methylation, the remaining processes have also been selected. So we are seeing a bit of a split here. From what I understood from Dr. Sripad's presentation also is the fact that methylation can lead to silencing of the genes, and this is one of the mechanisms which have been explained behind certain uniparental disomy syndromes such as Angelman as well as Prader-Willi syndrome. Yeah, so basically what Dr. Sripad has also written is that, only methylation of the histones is commonly associated with silencing of the genes. And acetylation of histones has been found in activated genes. So actually it leads to a proper abnormal activation of the genes in certain normal or abnormal activation of the genes. And phosphorylation has been found to be associated with DNA regulation, but it is uncertain as to whether it actually silences the genes or not. So I'm sure that you'll remember that methylation is associated with silencing of genes, and this may be beneficial or deleterious. Now, this is the next question. The commonest etiologies for cerebral palsy include all of the following except. Just enable the polling. This is a bit of a tricky one. Let's see the results. So the commonest etiologies for cerebral palsy include all of the following except. The majority of you has guessed it right. It's vitamin C deficiency, because you know that vitamin C deficiency commonly leads to scurvy, and in cerebral palsy children, if at all children are to be vitamin C deficient, it leads to epiphyseal dysplasias. And obviously, you are aware that the most common causes are prematurity, whereas a small subset can be due to cerebral hypoxemia, which is an intranatal cause, and hyperbilirubinemia, which is a postnatal cause. So when you're referring to cerebral palsy, you must remember that 80 to 90% of the cases are congenital cerebral palsies. That is 80 to 90%. Only about 10% are due to acquired brain insults which happen in the postnatal period. So I think this is, as I mentioned earlier, it can lead to epiphyseal separation, vitamin C deficiency, but it never causes cerebral palsy on its own, whereas the rest of the etiologies mentioned are associated with cerebral palsy. So this is the next question. Which of the following is not a prenatal cause of cerebral palsy? Please vote. Surprisingly, a majority of you feel that multiple pregnancies is not a prenatal cause. Now, what has been shown is that twin pregnancies and all, it definitely risks it leads to an increased risk of cerebral palsy in one of the offspring or both the offspring, because there's obviously a hemodynamic compromise which can be associated with the placenta when you have multiple pregnancies. And even usually, if you, in a multiparous female with increasing number of pregnancies, the risk of hemodynamic compromise has been found to be statistically more in many epidemiological studies. Now, CNS infection, I'm wondering, I agree that, you would consider TORCH, but TORCH, that is Toxoplasma, etc., cytomegalovirus or rubella, etc., to be causing cerebral palsy. But its actual mechanism has been found to be due to a transplacental hemodynamic mechanism of transport rather than the direct CNS infection. So when I'm referring to CNS infection, I'm referring to an acute encephalitis or meningitis, which is very often happens in the postnatal period. So by definition, this would not be a prenatal cause of cerebral palsy. So I did not specify the term torch here. So I'm sorry if some of you have misunderstood, but the question I got was CNS infection, which I think is referred to as a postnatal CNS infection, such as a meningoencephalitis. And torch per se, that is the congenital torch, etc., are not considered to be CNS infections de novo. They are more hemodynamic transplacental transmitted infections, which spread by a hematogenous route and involve multiple organs. And the brain can be only affected in a subset, but you might have just involvement of certain organs such as the heart or the eyes or the kidneys, etc. And these again are believed to have some epigenetic mechanisms for causing torch-related infections. So the correct answer is definitely CNS infections. So the rest of the etiologies are associated with uh, cerebral palsy. So I think this should be easy. Kernicterus usually causes what type of cerebral palsy? Does it cause an isolated spastic dystonic athetoid or a combination? I'm sorry, just took the wrong poll here. Please vote. Yeah, so most of you correctly answered that it causes both a combination of dystonic as well as athetotic cerebral palsy. So So you are aware of chronic bilirubin encephalopathy, which is due to hyperbilirubinemia, especially unconjugated hyperbilirubinemia in the postnatal period. And this can cause a profuse movement disorder, and usually it is diagnosed based upon the presence of encephalopathy in after 3 months of age in these children. That is, they have a static encephalopathy with associated movement disorder, which may be a dystonia or it may be an chorioathetosis. And more often it is a slow athetoid movement, axial as well as appendicular, which involves these children. And many of these children would have had a history of unconjugated hyperbilirubinemia. Often the bilirubin levels may be more than 20, but you can have it at a much lower level. Athetoid cerebral palsy with kernicterus has been associated even with bilirubin levels as low as 8 to 12 of unconjugated bilirubin. The chronic bilirubin encephalopathy should have a high index of suspicion in children who come to you or present to you as athetoid cerebral palsy. So again, this should be easy. What is the best treatment for localized or segmental spasticity? So the options are injection Botox, then tizanidine, dantrolene, and baclofen. So localized or segmental spasticity. Please vote now. I think we are developing a lot of mental fatigue, and I think we are all hypoglycemic. So I can also see the participant list coming down. People who are actually voting are limited now. But anyways, I am sure that all of you agree that it is Botox. And why not the other options? So what is the issue here now? Now, what we must remember is that, for more of spasticity, which involves the axial musculature, that is whether it is the trunk or the neck or even the bulbar musculature, tizanidine and dantrolene may be quite useful, and the baclofen is known to be specifically useful for spinal spasticity. But if it's a very localized or a segmental spasticity involving the cranial as well as the appendicular musculature, for example, if it is a craniocervical dystonia, obviously it is the local therapy with injection Botox, and this has been shown in many recent randomized double-blind studies to be extremely useful for localized and segmental spasticity, and this corroborates as level A evidence for the use of Botox in patients who have localized and segmental spasticity. This is another easy one. The genetic inheritance of tuberous sclerosis is please vote. So most of you have answered correctly that this is an autosomal dominant disorder. Now, this is very, very well known that both TSC1 as well as TSC2 are autosomal dominant disorders. I'm sure if you have attended the lecture, this should have been a simple one. Now, they have two different genetic loci, both TSC1 and TSC2. But what is also interesting is that, all the family members do not exhibit the phenotype because of this phenomenon, which is referred to as second hit phenomenon. So now the second hit phenomenon is also being used as in a separate terminology in the modern world of genetics in the form of somatic mosaicism. So many of the family members are somatic mosaics. They might have very subtle manifestations, which may be in the form of very subtle cutaneous manifestations, such as single or multiple cafe au lait spots or angiofibromas, of facial angiomas, and some of them may not have epilepsy or tumors in their brain, whereas some may be born without epilepsy and might be harboring silent tumors in their brain. So there is a significant variability of phenotypes. And what is also being increasingly recognized is the fact that certain children who develop tuberous sclerosis may be due to de novo variants. That is, these pathogenic variants are absent in their family members. So although this has an autosomal dominant pattern of inheritance, there is a possibility that the child's parents might be gonadal mosaics. That is, a variable proportion of the gametes are likely to carry the mutation. So although the parents may be unaffected, you might not be able to detect the genetic variant in the parents' blood samples. It is only when you examine the sperm of the father that you might actually detect the presence of this gonadal mosaicism. So this is also referred to as the gonosomal mosaicism because of the fact that the father, who might have a minor phenotype, might just be carrying a somatic variant inside the brain, and a significant proportion of gametes which carry the TSC1 or the TSC2 gene. And this is transmitted to the offspring, who subsequently develops frank features of tuberous sclerosis, both the neurocutaneous part of it as well as the epilepsy part of it. So remember that this autosomal dominant is, while in vogue, you must think about de novo inheritance as well as mosaicism when you are dealing with a child who has tuberous sclerosis. So what is true about subependymal giant cell astrocytomas, that is the SEGA, in tuberous sclerosis, except please vote. Yes, the hypoglycemia seems to be setting in even more into our audience now. So majority of you have answered correctly that the wrong statement is commonest presentation is rapidly evolving hydrocephalus leading to clinical worsening. So most of the SEGAs, as you understand, are indolent and they can have a very silent presentation, and it's only when you do the scans that you might detect these incidentally, and a small subset of them go on to develop as a rapid increase in size of the astrocytoma and subsequently obstruction and hydrocephalus. And uh, you must remember that when the patients, how frequently do you need to actually or do follow-up MRIs in these children? So in the early part, it is recommended that you do it every 2 years, and once you see that the SEGAs are stabilized, you can do it once in every 5 years. But it is possible that at a later age, some of the SEGAs might suddenly grow and become cause obstructive hydrocephalus. So, which of the following statements is true about the drug cannabidiol, which has recently been approved in both Canada as well as in Australia for treatment of epilepsy? Please vote now.
Dr. Menon?
Yeah,
Yeah, I think it's late, so we'll take this as the last question.
yeah, I guess so.
Keep our rest of the quiz as in another session.
Sure.
Yeah. We have a lot of questions, really. I think we have more than 15 more. We can have another session at the beginning as well, isn't it?
Yeah, I can send those questions to you. No problem. You can,
I'll
yeah,
let you do only,
yeah.
but let us see what they
So yeah,
answer.
so I think majority have answered that it is useful for seizure control. So the actual approval has come for its utility in terms of seizure control in patients with not only tuberous sclerosis but any difficult to treat epilepsy or epileptic encephalopathy. And it is not useful in metachromatic leukodystrophy because certain experimental therapies are only available in this condition. It does not reverse the process in metachromatic leukodystrophy, and it is not enzyme replacement therapy for patients with Fabry's disease. And it is not approved for use as a hypnotic, that I'm sure all of you are aware. Okay, so I think, yeah, I think we'll call it a day for today. And back to Dr. Shripad.
Yes, thank you. Thank you, Dr. Menon. I think we had a mixture of many more questions in our bank, and we will put up another quiz in the same area again in the near future. I hope this session was useful for all of you with the 2 lectures on genetics as well as a lot of neuro regression and neurometabolic disorder related discussions through the quiz. I thank all my speakers and my quiz master as well. And good night to all of you. Thank you very much.
Good night.
Thank you,
Thank you.
Manasi, as well. Thank you.
Thank you.
Thank you. Thank you.